drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. If you're getting value from this content, please consider becoming a paid subscriber to the Mind and Matter Substack. For just $5 a month, you'll get early access to episodes and other content, full episode transcripts, and I'll prioritize answering your questions in emails and the comments sections. You can also support me by checking out the links in the episode description to my affiliate partners who make various health and wellness products that I use and enjoy myself. You'll receive a discount and help support the podcast that way. And if you want the benefits of a paid subscription, but it's not in your budget, simply sign up for my free weekly newsletter, send me an email, and I'll give you a complimentary paid subscription. Word of mouth is the best way to help the podcast grow. So if you like what I'm doing, please share your favorite episodes with family and friends. The purpose of the podcast is to provide you with information obtained largely from primary sources, the people doing the research and producing new knowledge. This content is never meant to serve as medical advice. And with that, enjoy the episode. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.
Yeah, thanks for having me, Nick.
Can you tell everyone a little bit about who you are and what you do?
Sure. Yeah, my name's Dr. Iain Oswald. I am the research and development director here at Abstrax Tech, which is a cannabis research and development technology company here in California. And so we do things ranging from cannabis extraction all the way to flavor development for vapes and other types of products, beverages, those sort of things. We've actually moved even beyond cannabis now, looking into hops chemistry because they're very similar. And yeah, making some waves over in that field. But the majority of our work is in cannabis, and we try to publish research when we can on some of the cool stuff that we discover.
Yeah, and you've been working on things related to flavor and aroma. Can you give people just sort of a general picture of cannabis aroma in terms of, how variable is it? How important is it to consumers? How do people sort of in the cannabis industry or in this general realm think about cannabis aroma and why it might be important and where it comes from?
Sure. Yeah. So if we start kind of from the general public, so the really broad image, I'd say that a lot of people, especially non-consumers, associate cannabis specifically with that sort of skunky aroma that, it is kind of well known for. And, we published research on that, understanding that sort of chemistry back in 2021. And we talked about that in the past as well. But the truth is that there's much more to it than just that aroma. And why this diversity occurs is, it hasn't been well known. A lot of people within the industry, the scientific community of cannabis, has really focused on terpenes for the broader part of the last few decades. And they've done great work as far as kind of understanding that chemistry and how it might translate to maybe some of the effects of specific strains, as well as, kind of chemotyping as far as their aromatic qualities. But the nitty gritty is that there's much more to that. Yeah.
Mm-hmm. So most people who think about this stuff, when we think about terpenes, so these are volatile compounds that smell. They all have usually a distinct smell. They are common in the plant world. So, you see them in cannabis, you see them in fruits, you see them in vegetables. It's sort of an aromatic communication language for plants, you might say. And we know that, citrus fruits smell citrusy largely because of the presence of certain terpenes. We know that, other fruits, vegetables, plants, a lot of their aroma comes from specific terpenes or specific combinations of terpenes. Many of them are named for this reason. So, pinene is a terpene. You find it in pine needles. It gives them their piney smell. And so I think the presumption has been that, well, you see a lot of terpenes in cannabis, and therefore the aromatic diversity and the distinct smell that you get from different strains of cannabis are presumably coming from terpenes. But based on the research that you've done, that seems to be called into question to some extent.
Definitely. And the truth is this research is kind of following a very similar path to that of wine, as well as beer and hops and grapes. And we're kind of, cannabis is next in line, and understanding that, the most plentiful compounds that are volatile and give flavor may not be the most impactful as far as their odor characteristics or their flavor. And so, to your point, yeah, terpenes are found all over the place. They're ubiquitous in nature. I think there's over 400,000 described within the terpene class, which is ranging from monoterpenes to sesquiterpenes to then diterpenes and even larger things than that.
And I think it's thinking about abundance is logical. So after the cannabinoids, after the cannabinoids like THC, the most abundant things you tend to find in cannabis resin are the terpenes. And in parallel to that, we know that terpenes are aromatic. They are responsible for the smells that we smell in the plant world much of the time. And therefore, it makes perfect sense to think that they're going to be the primary drivers of these distinctive cannabis aromas that we'll talk about. But sort of what you just alluded to is that, the abundance is not necessarily a good index for importance. You can have highly, highly potent compounds that are present in tiny levels that actually drive a lot of flavor and aroma. Maybe a good hook into this for people that might be familiar is if you just think about cooking. Like if you cook food at home, it doesn't take very much garlic to have a very large impact on the smell and the flavor of whatever you have in the dish. And so sometimes the tiny ingredients have this outsized impact.
Yeah, definitely. And, we actually alluded to garlic in our initial paper reporting the sulfur containing species that make cannabis skunky for that very reason. And they are coincidentally kind of chemically very similar too, which is interesting. But yeah, the other component here is that, cannabis has been difficult to study, period, from a legal standpoint. So academics that might have more sophisticated techniques than other folks that are in the industry don't have access to it. Or if they do have access to it, they might not have access to what is representative of the most modern varieties on the marketplace, right? Yeah. And so that's another thing that I want to kind of point out here is that as the years have gone by, the decades, the probably centuries, the aroma of cannabis has changed throughout that time, right? And it may have been more dominant in terpenes, specifically in older varieties, than they are in more modern varieties. As people have bred for those specific flavor nuances, well, now we start to see other things besides terpenes becoming more important. And that's why on the marketplace, you can have things ranging from these very, very sweet varieties, which it'll be hard to find a terpene that has the aroma characteristics that would suit those. And on the other hand of the other side of the coin is there's also varieties of cannabis that are very savory now, which is really interesting because when people kind of think about, well, what is modern quote unquote exotic cannabis? A lot of people think of really sweet, but actually there's this whole other kind of side over there that is beyond what people would kind of call traditional sort of prototypical cannabis.
Mm-hmm. And so I guess by working at Abstrax in the private sector, you're sort of in a unique position, I guess, because on the one hand, you guys have the resources to use and implement, technical methods that are, cutting edge, basically, that you would get only in like a hardcore research lab. But on the other hand, you also have access to the real world cannabis that's out there being used by actual people, which a lot of academic research labs don't have access to. So you sort of have both pieces here.
Exactly. And to be honest, they're both necessary, right?
Mm-hmm.
Especially for the chemistry that we've been doing, the analytical chemistry, to find these really low concentration compounds. But I would almost argue that the source of cannabis that we're getting is actually just as important, like you just said. And I don't know if a lot of people realize this, but for a long time, the only way that people in an academic setting, such as a university, could conduct research on cannabis, they would have to go through, I think it was the University of Mississippi or Mississippi State, one of those two, where they actually had fields growing. And it was the only essentially legal way that people could grow cannabis and then analyze it. And not to say that, they were growing boof, if you will, but I am highly doubtful that what they were growing there was representative of what people were doing in California, over the last few decades. And
Mm-hmm.
there's actually been papers that have verified this,
Yeah.
that the varieties available to academics is not representative chemically to that on the marketplace. And
Mm-hmm.
so to your point, yes, I think we were perfectly positioned to, take advantage of our, this investment into really powerful analytical equipment, but also having access to some of the most exotic or, super just really unique smelling varieties out there to understand, well, why do they have those characteristics?
Mm-hmm. And so let's maybe talk briefly. So you've been on the podcast before. We've talked about this work. Let's just briefly summarize your last paper for people in terms of the major findings and to give them a sense for like how you're actually doing this. So what did you find before and how did you find it?
Sure. So the previous publication we did in 2023 was aimed at looking at essentially, well, what is the chemodiversity in modern cannabis across a wide range of varieties, right? We wanted to sample everything that we possibly could as far as aromatic characteristics. And so we worked with some cultivators in California, such as 710 Labs. They were actually co-authors on that manuscript. And they helped us kind of pick out like, yeah, these would be great varieties to pick. They have very unique characteristics. These other ones are completely different. And so we ended up procuring 31 ice hash rosin samples, which the reason we went with rosin, because people have asked, is because these compounds are in such low concentration that when we do our analysis, it's really hard to see them, period. But when you make a concentrate, such as rosin itself, you're concentrating all those analytes down, so it's easier to kind of see what is real and what's not down there near the noise level. And so the equipment we're specifically using is called two-dimensional gas chromatography. And this instrumentation essentially is a very powerful way of decomposing a complex mixture of compounds into data that we can actually go in and look at all of those individual components. And so we've tweaked our methods between using rosin and then also just our methodology internally to basically try to raise up the signal as much as possible so we can see what are these really low concentration compounds that might be existing that folks have ignored for a long time. And part of this was born out of your previous study, Nick, where in your work, you called out specifically these clusters, these terpene clusters. And I remember reading this and thinking, well, it's curious that there are varieties that I know have very different aroma characteristics that cluster into the same sort of, statistical grouping. And I thought this is great because it was lining up with what we were actually finding on our side here at Abstrax. And so in essence, what we did is we took all of these samples, we ran them on our instrumentation, and then we also did a sensory panel, meaning we took the human element, the human response to these samples. And so we got 7 folks from our team to systematically go through these samples and literally do a sniff test. What does this variety smell like, and describe it to us. And what we found was that, in essence, the terpenes that so many people in the industry have been focused on often will remain remarkably similar, even when the aroma characteristics are very different.
Hmm.
And that right there was kind of validation that, something a lot of people had been thinking in the industry. They've been calling out, yeah, there's probably these other chemicals in there, but there was never any direct evidence of which ones were most important, but then also which ones are there, period. And so during that study, during the analytical portion, using the chemical tools that we happened upon some really interesting new compounds that ended up driving the aroma characteristics of these very, very strongly. And so two of those that I'll call out are these tropical VSCs, tropical volatile sulfur compounds. And so these would be, again, kind of in that class of volatile sulfur compounds that the skunky gassy compounds are in, but they're chemically unique. Their
Hmm.
functionality is all specific to itself. And so this is the 3-mercaptohexyl functional group. And cannabis produces this in many varieties that are modern. It's in tangy, it's in mimosa, anything that has a very intense sort of citrus or grapefruit, um, a very tenacious sort of tropicalness to it. You're going to find it in those.
And so sulfur containing compounds. So you're not talking about a terpene here, but just sulfur containing compounds in general tend to be very potent in their smelliness, right?
Oh, very much so. Yeah, the human nose has adapted well to sulfur containing species, probably evolutionarily. A lot of things you probably shouldn't eat probably have sulfur, and that's the reason why we're so fine tuned to it.
And that's where
But
like literally the animal skunks that their perfumes contain some of these types of chemicals, right?
Oh, yeah, definitely. In fact, the primary component of skunk's aerosol is chemically identical to the major component in cannabis, which is prenylthiol. The only difference is cannabis has a double bond, a skunk doesn't. And so you can imagine, it's a symbol, but you're basically taking a few hydrogens away, right? Yet, that's why it has a slightly different aroma,
But it's
but
still
still very
skunky.
much
Yeah,
very, very
yeah, yeah.
skunky. Yeah.
So interesting. Okay, so basically, you guys are using instruments that are highly sensitive. They're more sensitive than what you would even see in a typical commercial cannabis lab. So
Oh, very
basically,
much so.
the most sensitive instruments you could use to do this type of work. You've applied this to cannabis products, to looking at concentrated versions of strains that are actually out in the real world being used by
Mhm.
people, and you have found that you can sort of map the aroma characteristics that come from humans. So you have people smell this stuff. This one smells sweet. This one smells savory, and so forth. And then you can actually correlate that very well to the underlying chemistry. And what are some of the major differences in the aromas? Is it like a sweet versus savory thing? How should people think about the aroma space here?
Yeah, so we were actually kind of struggling with how to partition these sort of aromas generally when we were first writing this manuscript. And when I was first kind of drawing it all up, and we were trying to think, well, how can we kind of understand this in a way that's relevant to a consumer but still makes sense. We were originally thinking of doing like an aroma wheel, but then when we were talking to some folks who were on the panel, they're like, well, the wheels are great, but they tend to be kind of recursive, and they come back, and you have two terms that might be kind of um, at odds against each other. And so, and even before that, when I originally was conceiving of kind of how to partition these varieties, I was originally just thinking, well, prototypical, maybe like OGs, Jack Herrera's, older varieties like that, and then just really sweet varieties. And so those would be these modern exotics like Skittles or Runts, those sort of things. But one particular user, when he was doing the panel, he was smelling GMO, which is a shorthand form for garlic, mushroom, onion. It's named that because it's literally so savory smelling that that's how they got their name. And he said, "Well, Iain, this isn't prototypical, and it's definitely not sweet." And so that's when I realized, you know what? Like, there's a savory side to cannabis as well that people, it hasn't really been called that specifically, but I thought, this is the perfect opportunity to, you know, correlate the chemistry we're talking about to these specific aromatic notes that don't fit into the other two. And
Hmm.
so our spectrum, our cannabis aroma spectrum is what we call it, goes from sweet to prototypical to then savory. And it really does partition out kind of everything that we've happened upon within that paper and within our lab really well. And we can literally map these onto the spectrum now. It's really cool because you can comparatively say, like analytically, like, yeah, this thing is to the human nose more sweet than this other thing.
Hmm.
I think that was a really cool novelty of the paper that we came
Hmm.
up with for sure.
Yeah, and so one of the things, that you mentioned earlier that's been a little awkward for people in the cannabis world to explain is, we think terpenes are responsible for the aroma. That makes logical sense. And yet, as you mentioned, when you look at the data that I and others have produced, when you just go into a grow facility and you know what the basic terpene profile is because they have to get this stuff tested, you often find that things with very similar, nearly identical terpene profiles nonetheless smell very different. And typically the response you get to that is, people kind of wave their hands and be like, well, the slightly different ratios of terpenes can do complicated things in our nose, and it's complicated. But actually, the answer, it seems, turns out to be much simpler, which is they smell different because it's other compounds that are actually accounting for the difference. Um, you also mentioned so two different strains, similar terpene profile, can have very different aromas. And that's because you've discovered that these other types of compounds that contain sulfur and things like that are actually what's driving the aroma difference, not so much the terpenes. There's also, not just comparing strains with similar terpene profiles, but very often in cannabis, people do what's called phenohunting. So they'll actually cross two plants together. They will produce offspring that are all very genetically related. They're all brothers and sisters, basically. And yet they can look very different, smell very different, and so forth. And so even plants from the same cross can smell very different, even though their genetics are highly similar. And I think that was something that got you to your next paper. So can you start to talk about that?
Certainly, yeah. So, the our first publication set the groundwork for like what is kind of the universe of chemistry for cannabis aside from terpenes, just on a broad perspective.
Mm-hmm.
But what we became interested in is, to your point, what does the chemistry look like, the diversity as far as genetically similar cannabis?
Mm-hmm.
And why is this important? Well, you hit the nail on the head. This is literally how people select new varieties, right? The reason why people select Baccio 41, Gelato 41, and Gelato 33 and not Gelato number 2 is because it had the characteristics, oftentimes growth characteristics, yes, but many people nowadays are selecting almost primarily for aroma,
Mm-hmm.
right? And looking for what smells the most unique. And so the fact that you can have, the same crossing, so the same parents produce, in our paper, we were just looking at 5. So that's
Mm-hmm.
not even a large sample size. Yet what we found is the chemical diversity there can be very specific that relates very well to the aroma characteristics. And guess what? The terpenes are almost identical. And this just
Mm-hmm.
goes to show that sure, chemically they are similar when you're not looking deeply, but there's a reason why people select this pheno over another. And if it's from an aromatic perspective, it's because of this really specific chemistry that we're talking about. And within that paper, lo and behold, it actually, one of the major components was so the samples were Starburst 36 from 710 Labs. There were 5 phenos from that. And the major differentiator ended up being essentially the tropical VSEs, which again contain sulfur, very highly odoractive. And their distribution correlated with other changes between the groups that were said to be more savory leaning versus more,
Mm-hmm.
straight citrus and
I
tropical.
see. So Starburst 36, you said, was the strain or the
Correct.
crossing. So you cross
Yes.
two
Yeah.
plants together. We're going to call everything produced from that Starburst 36.
Hmm.
But there's variety there. Just like, if people have big families, you've got brothers and sisters, even though you got the same parents, you have different heights, you have different hair colors and so forth. Um, so these plants are different, and then they're selecting. You looked at 5 different phenotypes, 5 different siblings, basically from that cross. And you're saying that some of them smell sweeter, some of them smell more savory. What was the aroma sort of differences? What did that look like?
So the major differences, 2 of the varieties were classified as being much more intense tropical sweet citrus. right? And so that would fall onto that sweet exotic side of the spectrum. Um, the other three were two of them were specifically much more on the savory side. And interestingly, descriptors such as cheese came up for those two. And I'll
Mm.
get to that in a second. The third one was kind of in the middle where it didn't have the same characteristics of either of them, but it kind of partitioned into the latter half, the little bit more on the savory side. And the cool part about that is that was chemically, but when we mapped that to the sensory panel that we did for this study as well, they partitioned out almost very, very well. Like it was very clear clustering of the chemistry that mapped onto the aromatic qualities of the samples.
Hmm. So some of these things smell sweeter, more of a tropical type smell. Some of
Mm-hmm.
them smell more savory or cheese-like. Then you've got this other one that smells, somewhere in the middle, let's say. Um, so naively, right? Naively, before, if you weren't familiar with your work, you might think, okay, the tropical ones probably have high limonene levels, and the savory ones probably have high levels of a different terpene. What were the terpene profiles that you actually found?
Yeah, so they were all very consistent. So they're all caryophyllene rich,
Hmm.
which is contrary to the paradigm that, if it's very citrus, it has to be very high in limonene,
Hmm.
right? That data right there, we have a graph to showing the top 10 terpenes, I think, for all the phenos, and they look very, very similar just in their bar plots. But that goes against everything that, people in the cannabis industry for marketing have been saying for the last 10 years, which is that yeah, I love these varieties because it has a lot of limonene because it has very strong citrus notes. Well, here we have five varieties, all have some amount of citrus, um, yet none of them have limonene as the dominant terpene. And so I think that was our first paper really showed that, but this to me was like kind of the nail in the coffin that limonene is, you know, fairly benign when it comes to the intense sort of citrus notes that people talk about in the context of cannabis.
Hmm. So just to summarize so far, Starburst 36 is the strain, but you're looking at five different phenotypes, five different siblings that come from the same cross. We're talking about plants that are genetically highly similar, but not necessarily identical to each other. They are grown under the same conditions. All of the processing of the stuff you're analyzing is done under identical conditions. So we're sort of controlling for some of the potential confounds that might come from those things. We get even though these are phenotypes of the same strain, we're getting some that are more sweet and tropical smelling, some that are more savory smelling, and the terpene profiles look similar between all of them. And so, uh, take us a little bit deeper. What were the specific compounds that were responsible for the differences between the more tropical smelling ones and the more savory smelling ones?
So there was a few main differences. The first was the more tropical varieties were had the greatest concentration of all of the what we call Tropicana sulfur compounds. These are these tropical VSCs. And these include threomercaptohexanol, threomercaptohexyl acetate, butyrate, and we actually newly reported in this manuscript the hexanoate. So there's a whole family of compounds that kind of fit within this sort of tropical passion fruit, grapefruit, heavy citrus sort of family of compounds within cannabis. And depending on those levels, you can get more of that or less of that.
Hmm.
And
So
what we found, yeah.
is it fair to say that there's a group of these Tropicana sulfur compounds that are like chemically related? They have a similar chemical makeup. Is it fair to say, based on your past work and this work, that if you sort of vary the levels of each of those, you're going to get something that's kind of tropical smelling, but one of them might be a little bit more citrusy, a little bit more grapefruity, and so forth.
Yeah, exactly. And a perfect example of that is our understanding of all this chemistry is, evolving all the time as we measure new samples. But what we have identified is the 3-mercaptohexyl butyrate and then the hexanoate tend to be much more citrus forward, so almost grapefruit like. Whereas the 3-mercaptohexanol and then the 3-mercaptohexyl acetate, they actually lean a bit more tropical, meaning that they present like passion fruit, maybe pineapple. Some of these they're a little, less defined as far as their kind of aroma characteristics.
And then what about the savory samples? What was the chemistry like there?
Yeah, and this is what was so cool. So I was actually one of the participants on the panel, and it was all blind coded and everything. And what was so cool about this was I remember distinctly after smelling one of these samples, and there was two that I called out to be cheesy, and those were the ones that partitioned the way towards savory. I remember smelling this and thinking, this smells like Stilton, Stilton cheese. I don't know if you're familiar, but it's a stinky blue cheese, and it was that sharp tenaciousness that it's not the same as cheddar cheese. It's not the same as a bit more mild cheeses. And when we go and look at the chemistry, it was those ones ended up having a much lower levels of these Tropicana sulfur compounds that make the tropical notes. But on top of that, we did identify some fatty acids, and these include octanoic acid and decanoic acid. Both of these are key ingredients in various cheese products, dairy products. They're ubiquitous in nature as well, but they're rarely described in the context of cannabis, and they're definitely not described in the context of aroma of cannabis. So that was really cool, and I will say it kind of validated my nose. I felt so that was fun.
I mean, it makes good logical sense, right? The savory cheese-like aroma is coming from something like a fatty acid, which is what you would expect.
Exactly. Yeah. I mean, decanoic acid. I think one of the descriptor terms people often give it is rancid, right? It's almost like a rancid butter or rancid cheese, just something that's kind of gone off. And all of this is, important when it comes. The concentrations are important here, right? They were in low concentrations, but if something has high odor activity, then again, just like the volatile sulfur compounds, it doesn't necessarily matter. The human nose can pick these up. So that was really neat. And the other thing, just from a chemotyping standpoint, and almost like an aroma typing standpoint here, was something that was very cool between the two phenotypes that were very tropical, and then the remaining three. The three that had less tropical notes, we detected, an appreciable amount relative to other samples of indole, which is a heterocycle, right? So indole, we previously reported in the paper in 2023 to kind of correlate more with the prototypical, definitely leaning towards the savory exotic side. And I thought that was fascinating that there's this sort of very specific and abrupt partitioning where it was like, I think it was the last figure in our paper where we actually circle indole into 3 chromatograms and highlight how it is very obvious in those 3 that lean prototypical savory and not in the other 2, which makes us start wondering, is there some sort of trade-off biologically, biosynthetically between indole and maybe some of these other metabolites that are important?
Yeah, I think that's likely. So we know cannabis consumers care about the sensory qualities of the products that they're interested in. They care about how it smells and how it tastes and so forth. And they're also interested in the effects, the psychoactive effects. And those two things may be or are plausibly related to each other. But ultimately, right, they want to know, how is this thing going to affect me? And before your research, I mean, even after your research to this day, the presumption is still that A, the aroma is dictated by terpenes, and B, the so-called entourage effect, which actually hasn't been demonstrated very clearly at all, in my opinion, between terpenes and cannabinoids. The idea is, the terpene profile is dictating the aroma and it's modulating the effect of THC. So by knowing the terpene profile, you can know the sensory qualities and the effects, and therefore we should be able to label products knowing these things, and that will predict how people will feel when they consume it. So the idea would simply be, let's just show people, say, the top 3 terpenes or something like that, or group them based on terpenes, and that is the appropriate way to label and categorize cannabis products. What does your research say about the potential effectiveness of that general approach?
I think, to your point, Nick, there's a lot of marketing around the entourage effect right now. And I think we're simplifying things down without really having hard evidence, at least not hard enough for myself as a scientist to really fully believe. But that's not to say that there is something to it. It's just we don't have the full chemical picture yet, right? And that's why I'm excited about this new research we're doing. Yes, these are in very low concentrations, but combined together, could they be doing something important biologically, physiologically. And something I'll mention that I do think is interesting that cannabis produces is, it does produce indole that people are, inhaling when they have these certain products. But it also produces scatol, which is in those extremely savory varieties. And the reason I bring these two up in particular is because they have these heterocyclic rings, and heterocycles are found ubiquitously in nature. They're a key core functionality, chemical functionality in tryptophan, in
Mm-hmm.
melatonin, in psychedelic drugs. That's not to say that they themselves have those characteristics, but it would be really interesting for us to now look at this new chemistry, especially the chemicals that kind of might have correlation to known compound classes out there, and maybe do some molecular dynamics or docking studies to see, does this have an affinity? Maybe indole has a slight affinity for the allosteric site on CB1, right?
Mm.
Maybe that influences how THC binds, those sort of things. But you can't do that unless you have the knowledge of what is there in the first place.
Mm-hmm.
And so I think that our research, I'm hoping other folks will take up, especially more on the medical or the kind of biological aspect, to see, do these things sort of modulate actual physiological responses? And if so, like maybe then we could start getting into tailor-made and truly accurate sort of effects-based predictions.
Mm-hmm. And so given that these things are present at such low levels, these tropicana sulfur compounds, these fatty acids, these other things that seem to be very important for the aromas that you've identified, they're present at very, very low levels, which is why you had to use concentrated samples in the form of hash rosin to do the study. Given that fact, and given the additional fact that most companies out there making cannabis products, most labs out there doing testing for the industry, they don't actually have the equipment that you guys have. They can't actually measure and detect these things. So is there any plausible way to actually use this type of information to communicate something to consumers about the aroma profile, given that no one except you basically can measure these things?
Yeah, and I mean that is definitely the struggle, right? Is okay, we've made these cool discoveries, but how can we bring this to the broader public? And so first is education, right? So that's the whole point of these peer-reviewed papers, is to educate other people, both scientists and people interested in cannabis science that might not be scientists, about this. But we are actively working on trying to translate some of this complexity onto less complex analytical systems. And so these would be things that people are using in typical analytical testing labs. And so we've had definitely some success there as far as certain compound classes go. I will mention that one of the benefits of the instrumentation we have that would translate fairly easily to a traditional gas chromatograph is this sulfur chemiluminescence detector. And so we've actually been developing this, and I gave a talk at AOEC last year about some of the research we've been doing, at least detecting the sulfur-containing compounds using this detector, because the only thing it detects is, does this compound have sulfur or not? So it simplifies the data greatly. And when you lose that second dimension that we have, the data is even more complex. So that detector has proven very valuable in the context of trying to develop a method that, if a testing lab just had this SCD, could potentially actually start reporting these very important compounds. And so then the other aspect is that I'll mention is reporting ppms, if you will, on a box, micrograms per milligram. I personally don't find it very conducive to be, or very useful in any way to understand, will I like this product or not?
Mm-hmm.
You can have very high concentrations of terpenes, like 4% on flower. They're typically terpinolene rich, which is funny, but they're not as loud as something that has less terpenes, because they're lacking those really low abundant potent VSCs that we're talking about. And so that could lead you astray, right?
Yeah. And so that sounds problematic from the standpoint of a proxy for quality, or at least aroma pungency, that's evolved in the industry is simply to look at the total terpene content, the presumption being more terpenes, more pungent, whatever the specific smell might be. It sounds like you're saying that's not going to work.
Yeah, I mean, or at least it could be refined greatly. Let's put it that way. And part of this again is education. if I did a class where I had a group of people and I had them sniff through the terpenes and then I had them sniff through these VSCs, I guarantee you every single one of them would say, "Oh, that's what I'm looking for. Those VSCs is what I'm looking for. Those other terpenes, like, oh, those just they kind of smell just like generic plants. They don't smell like cannabis."
Hmm.
And so I think that's a big part is translating this into something that people will find, not only interesting but useful. And so to that end, I mentioned having a percent on there. I don't think is very useful. But what we're doing internally is we're taking all this data we're acquiring internally, both on the GC, and we now do basically a sensory panel for almost every single sample that comes through our lab. We're going to take all this data and we're modeling it to look for relationships between the chemistry that we're already pretty well aware of what's going on, but can we create predictive models now that maybe from a scale of 0 to 100 be like, okay, yeah, this product, at least at this point in time, has a gas factor or a skunk factor of 95. It's going to be of a certain potency for that aromatic quality because it was analytically tested and fitted to this model. We could do that with potentially tangy, right, as far as the sort of tropical notes. And so that's what's so cool about all of this is that knowing this kind of subtle chemistry can allow us to actually devise metrics that are truly useful for the consumer if they care about aroma as far
Hmm.
as quality.
And how important is the time component here? So these things are very volatile. They float away very easily. They're present at low concentrations, and yet they have a huge effect on the perceived aroma. How does this relate to something like freshness? And if you've got a jar of cannabis at home, most people who've had the experience know that on day one versus day 5 versus day 10, that aroma does change. Are these things a good indicator of freshness?
Oh, incredibly. And in particular, the prenylated volatile sulfur compounds that we've discussed previously, they are the most volatile. Yet they are probably what most people are after when it comes to the quality indicator, through their nose. And just from a recent experience, we're doing a new study with some folks on some genetics, essentially. And I had the great luxury of harvesting some flower and curing it, and I don't have a fancy setup where I was, and so I had to use the brown bag method, and it was incredible how the aroma changes through that process. And I'm definitely not an expert, and I bet you on top of freshness, it's also probably a really great indicator of how good are you at your craft. Like
Mm.
if something gets to the store and it still smells very pungent, if somebody else like myself, a novice, was given the task of drying and curing and that whole process, there's no guarantee that it would come out the same.
Hmm.
And so I think actually that is actually a follow-up study we're working on right now, which is not necessarily the curing aspect. We unfortunately missed out on that, but once it's in the packaging, how does packaging affect this? Right. And so we're doing that right now where we're going to measure over the course of about 6 months. And so it should be really interesting because again, a lot of this I think people have suspected or they've known, but there's never been hard evidence to say, well, what is the rate of decay for these VSCs versus terpenes? And I think what we'll find is they're going to be much more rapid as far as their loss. And to your point, Nick, quality, that quality impact could be more rapidly lost. And so huge implications for people who care about quality and producers who care about quality.
Hmm. Yeah, and it's remarkable how good the human nose is actually when you start thinking about this. Like I have met many people in my personal life and in my time in the cannabis industry previously where they can tell you in a millisecond if something is truly fresh or it's been sitting out for a few days. They don't necessarily, they might not have any idea what they're smelling, what the chemistry is, but like they can tell you reliably and they're like, oh, that's fresh and that's been sitting out without being told that ahead of time.
Yeah, definitely. And I mean, I think that sort of training comes pretty quickly, to be honest. But again, you have to be exposed to that,
Mm-hmm.
right? And I think that's the major benefit. If you do home grow or you can get what is would be considered really high quality cannabis, you will know immediately, like, oh, this is different from everything else that I've had, right? And there's so many variables that go into the retention of these compounds. I'll mention, I did track one variety. I took 3 data points through this curing that I did 2 weeks ago. Again, just brown bag method, but it was fascinating because I could literally see there was a specific VSC that appeared, and I could smell it. It was because it was an OG, but it was beyond OG. It was much more savory. And it's not just prenylthiol. There was another one in there that leans a bit more on the savory side of kind of almost like truffles or garlic, to be honest. But as it cured, and again, I'm not an expert, but nonetheless, it changed to my nose and it went away fairly rapidly. And I thought that was really interesting for 2 reasons. One, does that mean if you want to try to capture that aroma and you're making concentrates, you want to harvest or maybe process at a specific point in time? But second, is there a way to maybe if I had been curing more correctly, if you will, could that have been retained more so it lasts beyond just the 2-3 weeks that it was kind of there? But it was really fascinating, and it was cool to validate chemically what I was smelling in the data.
Mm-hmm. Interesting. You mentioned some of the follow-up work you're doing. Is there anything that you want to reiterate about this last study or anything that we missed that you think is important that you guys found?
I mean, I think the biggest one is if you are a cannabis breeder, because I think, to be honest, the biggest story here, a lot of people have focused on the acids because implications for cheese aroma, and I think that's cool and all, but I think the broader picture here is if you're a breeder, using analytical techniques at testing labs may only lead you astray, or at the very least, it'll leave you confused. Because and I've seen this before on Instagram or social media where breeders will say, yeah, I had these 2 things tested. One smelled like this, one smelled like that, but the terps all look the same, and they're shaking their head. What's going on? And so I think analytics aren't there yet as far as really helping breeding, unless you're pheno hunting for new terpene profiles, which that's perfectly fine, right? It'll definitely be helpful there. But if you're looking for in the context of something where the terps look the same but smell different, then our paper really shows that it's going to be difficult to kind of do.
Yeah, and I think anyone pheno hunting for new terpene profiles, I don't think anyone is actually doing that for the sake of the terpene profile. I think they're doing it insofar as they believe it's going to tell them or dictate the aroma or the effects. But your work is really showing that could very well lead you astray if you're using a terpene-centric mindset to do that selection.
Yeah, no, exactly. I mean, that's literally what the main point of the story is that breeding is, it's still an art more than it is, I'd say, like a hard science as far as understanding these nuances. And the truth is, the nuance is what people are looking for, right? A lot of times these differences are subtle, but they're detectable, and it's how people make decisions. And so, I think just this sort of science just kind of helps kind of show us that we're not quite there yet as an industry to take advantage of some of this this new science, but hopefully in the near future it could get there.
Mm-hmm. Well, thank you for your time, Iain. This is fascinating stuff. You've been on before, so I'll just point people to, I've got another episode up with you where we get into some of the findings that you guys came out with last year to do with the skunky aroma and things like that. So these 2 episodes kind of go together in a complementary fashion. Anywhere, how do people find the study or find your work and that stuff?
Sure, yeah. So if you're looking directly for the peer-reviewed study, you can go to ACS Omega. So it's the American Chemical Society. It's a journal there, as well as you can go to abstraxtech.com, and we have a science section. And we have done white papers to kind of decompose all of this. There will be a forthcoming one for this phenotyping sort of this more modern work that we just did a few months ago. But yeah, if there's a lot of great resources on our website, so I recommend people checking those out. It kind of distills down some of this complex science into much more digestible sort of formats, especially if you're not, an analytical chemist like myself.
All right, Dr. Iain Oswald, thanks for your time.
Cool. Thank you, Nick.
This episode is supported supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.